very pleased to introduce our last presenter of the day. Biyu He was joining us from NYU. I have known of Biyu's work for many years, in part because of my frequent visits over to WashU, where Biyu performed her PhD work, which was really amazing work on spontaneous brain activity and the relation to neurophysiological signals. I know that BU has been working on issues related to the topic of the workshop really ever since, and has a lot to tell us about her own research now and using multimodal imaging techniques and computational modeling as well. So over to you, BU, and thank you for being with us today. 15-20 minutes, if we can keep it to that, that'd be great. And we look forward to your talk.
All right. Thanks so much, Olaf, for inviting me, and thanks, Randy, as well, for organizing and inviting me to this workshop. It's actually my first time participating in BCW, a very much long overdue attendance here. So I'm very excited. If I run over time, feel free to cut me off. So I'll try to stay within the allotted time. We've heard today from Giulio and Enzo about different states of awareness. And my lab has been studying a somewhat different question, also related to the topic of consciousness, which is what kind of neural activity during normal wakefulness generate different content of awareness. Specifically, we've been working in the visual system. So you could say that we've been tackling the question of what's the neural mechanism of conscious visual perception. Over the past few years, we've come to a set of tentative working hypotheses or interim conclusions, if you will. And I'd like to share with you what we found so far across several series of studies. First, across multiple paradigms and experiments, we found that conscious perception likely requires the coordination of large-scale brain dynamics across multiple brain networks. And in other words, conscious visual perception is not just generated by the visual network. And second, we found that conscious perception is strongly shaped by the pre-existing brain state, including both the active dynamics and the latent memories. What do I mean by that? So the active dynamics refers to the moment-to-moment spontaneous fluctuations in neural activity, and the latent memories refers to connectivity patterns that reflect past experiences. Today I'll take you through several series of experiments that led us to this set of tentative conclusions. And first, I'd like to tell you a bit about our approach. Our general approach is to take visual psychophysical paradigms that psychologists have developed over the past century. For example, these bistable figures, also called ambiguous figures, where the image has two different interpretations. And even when you fixate your eyes at the center of the image, your perceptual interpretation of the image, your conscious perception of the image can help but fluctuate, alternate spontaneously and semi-stochastically between the two different interpretations. So the figure on the right here, you may see it as a man playing a saxophone or a woman's face. And so once your brain knows the alternative interpretations, the same fixed stable stimulus can trigger this constantly changing conscious perceptual outcome. So we could ask what kind of neural activity support the different content of perceptual awareness. Another paradigm that we have used fruitfully is the threshold level perception paradigm, where a stimulus that's titrated to the individual subject's perceptual threshold, meaning that it's typically weak, in contrast, or brief in duration, or both, is repeatedly presented to the same subject. And it's sometimes perceived consciously and sometimes not. And a robust finding is that even when it misses conscious perception, it can still process by the brain to the extent of influencing behavior. And so we could ask, what is the difference in neural processing between the conscious and unconscious processing of the same stimulus? And lastly, more recently, we've been very interested in how past experiences and memories influence perception. We all know that different people can perceive the same stimulus differently, depending on their past experiences and individual dispositions. And if you have seen this picture in Psychology 101 textbook, the dog in the picture just pops out at you. It's a very automatic perceptual phenomenon. But before you recognize, before the first time you see it, before you recognize the dog in the picture, initially you may be presented with the picture multiple times and only seemingly blobs. So what's the difference in the brain? One that prior knowledge is able to guide your perception versus when you don't have the applicable prior knowledge. So the questions that we've been probing in terms of the neural mechanism, the paradigms, involve both the difference between seen and unseen files, but also very importantly, we've been focusing on the neural encoding of conscious content. What kind of neural activity gives rise to different content of experience, and we've been employing a multimodal approach to look at both spatial representations and neurodynamics with both spatial and temporal resolution that's offered by fMRI and MEG. So I'd like to first tell you a bit about what we found with bistable perception. So here, the first experiment we did was to present subjects with these classic bistable perception pictures, and subject report their perceptual outcome as they fluctuate over time. And we simply asked previous studies have only looked at visual cortex, and here we asked, if we open up the lens and look across the whole brain, where in the brain can we use the local activity pattern to decode the instantaneous perceptual content experienced by a subject? And as a control, we also modified the images to reduce the level of ambiguity so that the image is typically perceived as looking down on the cube or looking up at it, or two faces and a base. Whereas these bistable figures, ambiguous figures, trigger constantly fluctuating perceptual outcome. And we found that in visual cortex, for example, in V1, we could decode the content of conscious perception both when the stimulus is ambiguous and when the stimulus was unambiguous. However, when the ambiguity in the stimulus increases, which I think is a very important model for studying natural vision, because stimuli in the natural environment often contains a lot of ambiguity due to occlusion, clutter, inherent complexity of natural objects, lighting. And here we found that when the stimulus contains ambiguity, we could decode the content of perception in ventrofrontal cortex. But in these higher order regions, we could not decode when the stimulus was unambiguous. So we think this additional recruitment of higher order regions to represent perceptual content is potentially consistent with recent neurophysiological findings from Jim DiCarlo's lab. And the next thing we asked is, what's the information flow across these different regions? Here we use Granger causality. I'm not going to go into the details of the method, but just to mention that one caveat with applying Granger causality to fMRI, which is well known, is that you may have heterogeneous HRF hemodynamic delay across different brain regions. And to circumvent that problem, we exclusively probed changes in Granger causality patterns between task conditions, because it's known that the hemodynamic response function for a region is going to stay constant regardless of what the task condition the subject is involved in. And we extracted our eyes across the brain, and here what I'm going to show you is that they are sorted from more posterior to the more anterior regions in both x and y axis, and each element tells us the changes in Granger causality influence from a source ROI to a sink ROI between the two different perceptual conditions, simple and bistable perception. And the bottom triangle is purely bottom up, and top triangle is purely top down. And we found that when a stimulus contains ambiguity, there is dramatically enhanced Granger causality across the entire network, both in the bottom-up direction and in the top-down direction, suggesting that to resolve perceptual ambiguity, you both need to involve frontal regions to represent perceptual content, you also need to elicit, involve a lot of bottom-up and top-down bidirectional information flow across the large-scale network. One neat thing about this paradigm is that if you present these images intermittently, the perceptual switching rate dramatically slows down, even to the extent of being brought to a standstill. What this means is that during these gaps, when the subject just sees a blank screen, there is a perceptual memory trace such that the most recently experienced percept is more likely to be reinstated when the image is shown again. So we tried to decode this perceptual memory trace, and we found that it's exclusively encoded in higher order regions, and it's accompanied by a strongly enhanced top-down input from frontal to ventral visual stream, and at the same time reduced bottom-up input, suggesting that perceptual memory, which we think is a phenomenon that may be important for maintaining perceptual continuity during natural vision, involves higher order regions and increased top-down input. So next, I'd like to dive into a series of studies we did with threshold level perception, where we probed what's the difference between conscious and unconscious processing of the same visual stimulus, and how is the conscious content represented in the brain. The basic paradigm is something like this. Subject fixes at the cross, and after a random delay of a blank interval, a stimulus comes on. Here is a ball patch pointing to upper left or upper right with equal chance on any given trial. And then after another delay, random delay, subject is cued to first answer about the orientation of the ball patch, a false discrimination task. And then they report their subjective awareness. Did you consciously perceive the stimulus or not? And we titrated the stimulus to the individual's perceptual threshold. So they answer same in about half of the trials. And as I mentioned earlier, when they report seeing the stimulus, their discrimination performance is about always correct. But when they report I don't see the stimulus, the discrimination performance is substantially lower, but still above chance level, suggesting that the unseen stimulus nevertheless influenced discrimination behavior. We recorded MEG while subjects performed this task. And just to foreshadow what's coming up, we found that the low frequency MEG signal in the less than 5 Hz, which is part of the broadband aperiodic activity, is the strongest correlate of conscious perception at a single trial level. So what we did was that the stimulus was very brief, about 60 milliseconds, and using neural activity, large-scale neural activity, at every time point from 2 seconds before stimulus onset to 3 seconds after stimulus onset, we asked, could we decode the perceptual outcome that is going to be reported by the subject much later? And in fact, we could, using the ICP activity, just low-pass filter data at less than 5 Hz, we could decode very well up to 2 seconds before stimulus onset, which is not shown here. And that's shown in magenta. And after stimulus input at the single trial level, we could decode conscious perceptual outcome with about 80% accuracy. And at the single subject level, you can see this effect for every single subject. And what's contributing to the decoder, it's really large-scale activity across frontal occipitotemporal areas. This is the topography of MEG sensors looking down on the brain. The nose is up here, left and right ear here. And you see that pre-stimulus activity has strong contribution from frontal cortex. And after stimulus input, occipitotemporal and then later on frontal activity contributes most to differentiate between seen and unseen trials. So next, we wanted to know, have a more intuitive sense of what is the dynamical signature of this large-scale brain activity that underlies conscious visual perception. We used dimensionality reduction to project the high-dimensional MEG dataset to a reduced low-dimensional space. Here we used good old PCA, and the top 5 PCs accounted for more than 17% of the variance in the data. And plotting a single subject's data from 1 second before to 3 seconds after stimulus input, we find that for pretty much every single subject, we find well-separated trajectories in a state space between seen and unseen trials. And importantly, these trajectories are separate even before a stimulus input. So even before stimulus input, they reside in different locations in the state space. And depending on this initial location, the same stimulus triggers the brain to travel on two different trajectories that correspond to seeing the stimulus versus not seeing the stimulus. And one important finding here was that in the same trials, the neural activity goes through a sequence of activity patterns that's fast changing over time, which is indicative of what's called transient neurodynamics in the theoretical literature. And another important dynamical characteristic is that it's accompanied by strong across-trial variability reduction, suggesting that these transient dynamics, even though they are fast changing, they are robust to small variations in the initial state, are robust to noise. So together, these results suggest that large-scale brain dynamics underlying conscious vision can be described as initial state-dependent, robust, transient neurodynamics. And in a later experiment, we dig deeper into what kind of sources contribute to this spontaneous activity in the pre-stimulus period that can influence conscious perception versus failure thereof. So some background here, it's well known that spontaneous activity occurs at multiple different spatial temporal scales, but how spontaneous activity at these different spatial temporal scales influence perception and behavior, I think at the moment remains a big unknown. So in this particular paradigm, we asked, can we use what kind of neural activity in the pre-stimulus period influence perception? Specifically, how do they influence perception in a content-specific manner or non-content-specific manner? Do they change perceptual sensitivity or criterion? And we basically use the same paradigm as before, except that now to get a better sense of how what's the content encoding in the neural activity, we used instead of low-level Gabor patches, real-world meaningful object stimuli that could belong to one of four categories. We also included scrambled images that preserved low-level image features but do not contain any meaningful content. So this allowed us to analyze the data in signal detection framework, where for real images, say recognized versus unrecognized would correspond to hate versus miss. For scrambled images, they would correspond to false alarm and correct rejection. And again, the stimuli were titrated to the perceptual threshold, so real images were recognized in 50% of the trials. Importantly, a substantial fraction of scrambled images were reported as recognized, which correspond to false alarms or false perceptions. That's when subjects perceive a meaningful content out of meaningless noise. And we think it's a very interesting phenomenon and a potential model for understanding visual hallucinations. So the take-home message from this study was that we found two different spontaneous activities that influence visual perception in different manners. One process we call non-content specific process, it correlates with pupil-linked arousal. And when this activity is high, subjects are more likely to report recognition regardless of what's the stimulus input. And it does so by shifting the detection criterion or decision criterion into the more liberal direction. And another process, which we call content-specific process, has different activity patterns that specifically enhance recognition for different types of stimuli. It's not correlated with arousal, and it influences recognition by changing perceptual sensitivity. So I don't have time to go into the data here, and if you're interested, you could read the paper here. But I do want to show you a follow-up fMRI study that just came out recently to understand what is the neural activity triggered by the stimulus that correspond to conscious versus unconscious processing of the same visual object stimulus. So we use the same paradigm, except now we recorded 70 fMRI, and the timing was adjusted to fit with fMRI. And the first shock that came out of the data here was that the spatial extent of activation and deactivation was nearly identical between seen and unseen trials, between the recognized and unrecognized trials. In both cases, we found widespread activation across the visual and task positive network and deactivation in default network with almost the same spatial extent. And in subcortical regions, also the same regions, thalamus, basal ganglia, and brainstem nuclei were activated and deactivated in both recognized and unrecognized trials. So this goes against some prevailing theories about consciousness that proposes that conscious perception happens when additional cortical areas are recruited. So what's different between these two cases? The first thing we found is that in recognized trials, the activated regions are more activated and the deactivated regions are more deactivated. So there was a quantitative differences here. In subcortical regions, basal ganglia, certain brainstem nuclei, I'm not going into the details there, and thalamus, we also found enhanced activation with recognition. And so that's the first difference. And the second difference is that across almost all of these activated and deactivated regions, we could decode the perceptual content in recognized trials, importantly in both activated and deactivated brain regions. So in other words, across visual network, task positive network, and default mode network, we could decode the content of conscious recognition. The decoding of stimulus category was at chance level for unrecognized trials. However, in subcortical regions, we could not decode any content. So what it suggests is that you have content representation across widespread cortical networks, both activated and deactivated, accompanied by stronger non-content specific activity in subcortical regions. So broadly speaking, we think this may be consistent with what Mike Lasser just showed us, where you have more compressed thalamic signals accompanied by content-specific cortical signals. And we think that the subcortical regions may facilitate the emergence of content-specific activity in cortical regions. And we also had some interesting findings here about what drives the false perceptions in scrambled images. And just to give you a quick summary there, we found that maybe default regions together with anterior cingulate and inferior frontal junction are involved in top-down inference during viewing of scrambled images that drives false perceptions. So lastly, I want to quickly show you some results about memory's influence on perception. So we are fascinated by this Dalmatian dog picture kind of phenomenon, which really tells us that a simple experience can change perception in a long-lasting manner. So we recreated this phenomenon in the laboratory with these images that are degraded. For many of you, you may not recognize what's in this picture, but now if I show you this picture, for at least a large fraction of you, you will see a dog out of this picture. And this effect is very long-lasting, very robust. It's called one-shot perceptual learning. And to first show you the behavior pattern in this task, we presented subjects with a set of these morning images 6 times before and 6 times after seeing the corresponding grayscale picture. You can see that percent recognized increased a little bit across the repeated representations, but there's a big jump after seeing the corresponding grayscale picture. That's the disambiguation phenomenon. And we also included catch images where the grayscale did not match, which controls very robustly for repetition phenomenon. And there is no, repetition effect is much smaller. And this is also consistent with verbal identification accuracy, a different way to measure image recognition. And a very recent study we did in collaboration with Larry Square's lab shows that this one-shot perceptual learning phenomenon does not depend on hippocampus or the medial temporal lobe or the conscious memory or episodic memory, declarative memory system. So this is data from amnesic patients with medial temporal lobe lesion, and it's as good as controls, and also shows that the learning effect very long-lasting, lasting for many months. So what's the neural substrate for this one-shot perceptual learning? We don't have the full answer yet, but we have some intriguing findings in the cortex that may give us a clue about what's happening. So to understand how the cortical representation, neural representation for single images are changed after acquiring the relevant prior knowledge, we use representation similarity analysis. So in this task, we have 33 different morning images. We can land them up here from number 1 to number 33 in the pre-disambiguation stage, in the post-disambiguation stage, and in the grayscale stage. And every element here tells us how differently a brain region represents two different images, for example, in the pre-disambiguation stage, or between different images across perceptual stages in these squares. And when we look at primary visual cortex, we get something like this. There are two salient patterns here. First is that this post square is redder than the pre square. What that means is that in V1, different images are represented more distinctly after disambiguation. After you have the prior knowledge, different images are more differentiable. The second pattern is that you see blue diagonals here and here. That's the same picture presented before and after disambiguation, and this one shows that the same picture presented post disambiguation is represented more similarly as its matching grayscale picture, and we can quantify this effect, and it's all very significant. And the biggest surprise in this study came when we looked into higher order regions in front of parietal and default mode network, where we found similar effect but even stronger. For example, in posterior cingulate cortex, which is not thought to be involved in visual perceptual processing classically, we found that the pre-disambiguation pictures are represented very similarly, and after disambiguation, the same black and white pictures are represented more distinctly, showing yellow and green here, and more similar to the matching grayscale picture. We have a recent paper that digs deeper into what really happens in these regions, and we find a gradient of sharpening effect across this entire cortical hierarchy. And lastly, we wanted to know what's the neurodynamics underlying this phenomenon. A long-standing puzzle about this one-shot learning phenomenon is that recognition-related reaction time is very slow. It's in the order of 800 milliseconds to 1 second. Even after disambiguation, recognized pictures has a RT of 800 milliseconds. And this suggests that long-distance recurrent cortical dynamics are probably involved. And we asked, can we find the corresponding neural signature with MEG? Here we took we basically did something like single trial decoding between the same picture presented at different stages of between Moni and matching grayscale pictures. And what we found is that first, if you look at the difference between pre-disambiguation and grayscale picture, this is one you have different physical image input, different recognition outcome, not recognize meaningless blobs versus recognizing a crab. You'll see that the neural distance between them shoots up after stimulus input and stays high throughout image presentation. But if you look at the Moni, the same Moni picture presented before and after disambiguation, you find that their neural distance increases after stimulus input and reaches significance around 300 milliseconds and stays high. And lastly, this is the most intriguing. When you compare a post-disambiguation Moni picture and the corresponding grayscale picture, different image input, different sensory input, but similar recognition outcome. You see a crab in both cases, because here you have the aid of the prior knowledge. You find that they're initially distinguishable, but while distinguishable within first 500 milliseconds, and then after 500 milliseconds, in terms of large-scale brain dynamics, they are indistinguishable. So this relatively slow timescale gave us a clue about the long-distance recurrent dynamics involved in solving this task. And we probed it more explicitly by combining MEG and fMRI, which allows us to map spatial-temporally neural activity related to stimulus processing and recognition content-related processing. And we found that the former is related to a feed-forward sweep that onsets first in V1, then V2, and V4, and then to higher order visual regions and from the prouder regions. Whereas recognition-related activity onsets first in higher order regions, in higher level visual areas in front of parietal and default network, and then moves back to early visual areas. So together, this last part of the talk tells us that one-shot perceptual learning and memory-guided perception requires recruitment of large-scale neurodynamics across multiple networks. And I think it's also very important for understanding natural vision, because in the natural environment, our memories are constantly guiding our perception. So to summarize, we found that conscious perception likely requires a coordination of large-scale brain dynamics across multiple brain networks, and it's strongly shaped by pre-existing brain state, including both active dynamics manifesting in spontaneous activity and also latent memories, which reflect your past experiences. And last but not least, the most important slide is to thank the people who did the work and my funding sources. And we have a postdoc position starting in the fall. So if anyone is interested, please feel free to get in touch with me.
Very cool work, Biyu. Wonderful talk, showing us the benefits of combining different techniques, different modalities, and all of that, combining it with modeling. This is fabulous. Reactions from the audience or from the panel. I am looking around. I see Randy has his little blue hand up. Very good. Over to you, Randy. Your hand is blue on my screen. Sorry.
No,
it's not.
It doesn't look blue on my screen.
Yeah, it's looking, well, anyway, it's okay. It's not blue in real life, we hope. Anyway, go ahead.
Could be. Anyway, thank you, Biju. It was great coverage of all those 3 areas of work. I must admit, I missed some of the stuff because there's a lot of information packed in there, but I think I got the gist of what was going on. One of the questions I have with a lot of this sort of line of work is being able to differentiate the preparation for motor response from the actual, we'll call it the cognitive process or the perceptual process that underlies consciousness. And I know that, you know, in some of the work that I've done on perceptual, this kind of work, it's very difficult to differentiate those two because it's so sort of ingrained in how people operate. So I wonder if you have, like, how did you control for that, or what are your thoughts on that issue?
Yeah, so for motor, that's relatively easy to control. So we could randomize perception to response mapping in some of the tasks. In the last MONI image task, we are looking at the neural activity pattern related to individual images, where all of the recognized images have the same button press. So the motor response for all of the recognized images is exactly the same. And I think that you know, even stronger control is what we did in the second part of the study, where in the fMRI part we could decode the stimulus category in recognized trials across all of these brain regions. But in unrecognized trials, we could not decode either the stimulus category or the reported category. In unrecognized trials, subjects were also asked to report you know, the category with their best guess, and reported category was completely undecodable, as was stimulus category. That means that the low-level image features was not driving the decoding, and the motor response or decisions was not driving the decoding.
I'm thinking like, especially in the bistable perception part, where there is, you know, it's either you see it or you don't, and in that particular, you see the switches, I should say. And if you know, the recruitment of frontal areas in that context, it has to do with actually preparing the response more so than the actual switching of the percepts per se.
Yeah, so in that initial study, we had very similar response requirements for the simple perception and bistable perception case. So if you see, for example, the top-down view of the cube, you press one button, and for the other view, you press the other button. And we could not decode in frontal cortex in simple perception, where the response demand was the same. So we think that it's probably related to increased perceptual ambiguity and the need to involve ventrofrontal cortex, which is only one synapse away from IP, to solve perception in that case. And it also resonates very well with work by Fanny's and Nicola studies groups, where in monkeys they used it's a related but somewhat different paradigm, binocular flash suppression, where they could decode, right, so it's like decoding. So basically the neurons in prefrontal cortex track the perceptual content even when the monkey was passive fixating. And more recently, they used a no report paradigm that removed reporting requirement, but used eye movement to infer the perceptual content, and we got similar result in PFC. And here we could not use no report paradigm because we wanted to keep it naturalistic.
Sure.
By inducing vestibular perception using ambiguous figures rather than binocular rivalry or binocular flash suppression.
Yeah, I mean, this kind of gets back to, you know, the stuff that Julia was talking about in his presentation in terms of what area. I mean, this may not be exactly the same, so we have to be very careful to not make the connection between Julia's notions about what is important for consciousness first versus what is actually important for working with the contents of consciousness, which might be something different. So, for example, I don't know if the results in perceptual awareness, for example, or detection, where the frontal lobes actually recruited, is inconsistent with what Julia was saying, or is that just sort of the consequence of having this very connected system that's dealing with the contents of consciousness and then having to do something with it after it's actually part of that full experience?
Yes, I would say that across multiple experiments, we do not see the division between frontal and parietal cortex as postulated in Julia's talk, but what we see is more of a gradient across large-scale interconnected networks. What I mean by that is that we see a difference between, for example, the frontal parietal network and the default network, and both of which have, you know, frontal and parietal nodes. But the frontal areas and lateral parietal areas that belong to the FPN, frontal parietal, also called multiple demand network, or executive control network, they are much more similar. And then the medial frontal cortex and the medial parietal region that belongs to the default network behave much more similar. So and which makes sense. I think, you know, it's also consistent with recent large-scale cortical gradient work by Daniel Margulis and colleagues. In fact, in the MONI task, if we look at the representation geometry of these different regions and how they differ across all of the regions we look at, we can recover the same hierarchy that Daniel has described, where you have, you know, early visual regions, high-level category selective visual regions, then FPN from the parietal network, then DMN. So it's you know, it's really beautiful correspondence between a task-related neural hierarchy and the resting state hierarchy, network hierarchy.
Great. I have another question, but maybe I can pass it off for now, Noel, and come back.
Yes, we'll come back to you. We have a question from the audience, Karim Jarbi. If you're out in the audience, please raise your hand. There you go. You got the mic.
Okay, thank you so much. Thanks, Biyu, for this wonderful talk. Nice to see you.
Hi, Karim.
I have a quick question related to the beautiful one-shot learning study, specifically in MEG. So I was wondering there, in terms of the features that you looked at, did you see like some of this like previously reported gamma oscillations that showed in the past, like some effects between seeing the object after I know what I'm looking for and seeing it before, and also the connectivity between areas? Is that something that you looked at also in the MEG data?
Yeah, we did not look at that. So that result is showing the difference between recognition and no recognition. And in recognition, if you recognize the picture, you have stronger synchrony, gamma synchrony across different regions. We didn't look at that partly because we wanted to focus on the representation of content. How does the neural activity represent recognizing a crab versus a motorcycle versus, you know, something else? And the contrast between recognized and unrecognized has you know, multiple processes that could contribute to it. Like when you recognize something, you know, you pay more attention, there's more salience, and you know, all of these are, some of them could be, you know, very integral part of conscious perception or immediate consequence that's, you know, hard or impossible to dissociate. But here we wanted to understand the content representation. And from earlier studies in our lab, we found that when we try to decode the content, really the low frequency signal, which contributes most to the classic ERPs and ERFs, encodes most of the content. And if we filter in high frequency range using either the filtered signal or the amplitude, sometimes we could decode, but the accuracy was a lot lower.
Right, and that's interesting because, as you know, there's been some debates about the origin of some of the gamma related in visual tasks, the gamma oscillations, and whether they relate somehow to miniature saccades or they were artifacts of eye movements. There was a whole literature on that, and so it would be interesting to see how much replication we see specifically in the gamma range. And as you say, alpha and beta makes more sense for the low frequency.
Yeah, absolutely. It's probably a stronger problem for SCOP EEG than for MEG, but nevertheless, we need to be careful about that. Yeah.
Right, absolutely. Thank you so much.
Okay, thank you very much for that question and the discussion. Back to you, Randy.
I think Mike had a question. If I'm not mistaken.
So Mike Halassa, if you can, are you on? Because you had a comment in the chat and a question attached to the comment. I wasn't sure who the question was for. Was it as well? So if you want to revisit that, go ahead.
Sure. Yeah. I thought it was an extremely thought-provoking and an amazing talk, Biyu. Thank you. I was wondering, I'm trying to sort of understand your findings from the perspective of ignition and some of the theories about what PFC does. So you were clear that you just don't see that's not really a big component of what you've been observing. This engagement of the frontal cortex that's specific to conscious perception, but there were these signal differences. Were those frontal? And then there was something intriguing, which is that you were saying that the signals that the areas that show deactivation also had a higher decoding accuracy on the labels of the objects that were presented, despite the fact that their activation was less. Is that true? Did I get that or no?
Can you see my screen? Am I sharing again?
Yeah.
Great. So so about the first part of your question, that's right. So that part does seem to be inconsistent with GNW theory, which at least earlier versions postulate that the signature of ignition on conscious perception is recruiting of the global workspace and the higher order frontal parietal regions. But there have been, a trickling of, a series of studies showing that unconscious processing can penetrate PFC, and that has been incorporated into more recent versions of GNW theory, like in the most recent neuron review. But I think, nevertheless, this result is shocking because it shows that conscious recognition and unrecognition, non-conscious, lack of conscious recognition, activates and deactivates the same large-scale networks. And we spelled that out in the paper. And then the second part of the finding here is that in the recognitions, when the stimulus is recognized, consciously recognized, you have enhanced activation and enhanced deactivation. And that's a quantitative difference between conscious recognition and failure thereof. And there is also a qualitative difference, which is that in recognized trials, the stimulus category, which matches nearly 100% with recognition content, recognition category is decodable from all of the networks in all of the in most of the activated and deactivated regions. So anytime you see a circle around it, sorry, I didn't point this out in the top, the circle around the dot means that there is significant decoding. And so it's in most of the deactivated regions and most of the activated regions, with a substantial salient gap of the subcortical regions where we could not decode.
Why is chance at 25%?
Because there are 4 categories.
I see. I got it. Okay, cool.
Yeah, so this DMN finding is, I think, is very important because although different theories have postulated that default network, given its central hub connectivity, may contribute strongly to conscious perception. But I think in theories about conscious perception, I guess they've been postulated that it contributes strongly to conscious state, being able to be in the conscious state. But for theories about conscious perception, the DMN is often ignored. And so seeing that we have, decoding of conscious content. And in the scrambled images, the false perceptions trigger stronger deactivation of the MN along with content encoding that corresponds to the perceptual content, suggest that this network may be really important for conscious perception. But I think we have a lot of questions that we need to work out still.
That's great. Thank you.
I was fascinated by your comment about the basal ganglion. I'll have to talk to you more about that later.
Yeah, we can talk about definitely.
Very cool. Very cool. I'm looking at the panel. Randy, maybe if you want to take it over for just a minute, I'm looking in the chat.
Biyu,
just one thing, those of you who are wanting to ask questions, I would prefer, and also for maybe tomorrow, the next session after that, do use the Q&A box. So there's too many things to track otherwise. I'm never quite sure whether you're asking each other questions in the chat or whether you're asking a question to the attendee. So if you have a question for the speaker, then please put it in the Q&A. Over to you, Randy.
Good. Biyu, since you had the slides up, can you pull up the one that had the PCA trajectories?
Yeah, this one.
That's great. I was just curious about that. I'm not sure if you were there for Victor's talk on the first day or not, but
I missed it, sorry.
so there is this notion that some of the trajectories that once you get sort of, if you're on a, we'll say a manifold that has attractors that control a flow across the manifold, that once you're in that that flow, it's almost deterministic. So you start at that place, if it gets activated, you actually have a very sort of deterministic trajectory that you follow every time. And I'm just curious, like I got the impression that this is amalgamated data, or is this a group average or something like that?
It's a single subject data. So, but we see similar phenomena in every subject.
So because it does look quite nice in the sense you said it's very sort of like once they're in there, they have that very sort of characteristic flow through. In this particular case, you've got a.
Yeah, I think the data that speaks most strongly to that is the across trial variability reduction. So on the left here, I didn't include it here, but it's in the supplementary of the paper where we showed single trial trajectories, and you can see at the single trial level, the trajectories are separated between syn and unsyn cases. And this is trial average trajectory for the syn condition, the two different stimulus orientations, and for the unsyn condition. And then the syn condition is accompanied by strong variability reduction.
Yeah.
That does suggest something, in the spirit, along the spirit that you were saying, deterministic. I hesitate to use the word deterministic because that's a pretty strong word here, and I don't think we really tested that.
Well, I mean, it's more. I'm not sure. I'm saying that it's deterministic in the sense that you can't. You're kind of stuck in a stereotypical mode, but it's kind of like if you're taking a step, it's hard to stop that flow from taking a step. So you kind of get stuck in.
Right. So we summarize that in the robust terminology here. By robust, we mean exactly that. So if we, I don't have the summary figure here, but our intuition here is the picture view of this is that you have a tube here that kind of narrows after stimulus onset, and you can, in the vicinity of this initial state, you get attracted onto it, and then you get in the vicinity of a different trajectory, you get attracted onto a different trajectory that corresponds to different perceptual outcome.
Sure. So that would actually then, if that's true, then that could be another. Maybe, I mean, I guess that's not necessarily independent, but that could be a explanation for why you have reduced variability then.
Yes, yes.
Because if it's a deterministic trajectory, then you'd have less variability, which gets pulled into that sort of flow, I would guess.
That's right. That's right. So it's we were very much inspired by work by people like Rabinovich and others that describe these robust transients in computational models. And I think the data is quite consistent with those models. And one thing that I think our data add is that we put this initial state dependence in the context of the same perceptual task, very front and center in this, in these representations. But I think it's completely compatible with those.
Cool. Thank you. I think you got some questions from the audience there, Olaf.
Yes, there's one from Dan Lurie. Dan, once again, let's raise your hand and, or rather you raise your hand and I'll unmute you. Let me see here. There you go.
Yeah, thanks. This was a super interesting talk. I was wondering, so given that you're kind of describing two kind of broad classes of influence of spontaneous activity on the perceptual awareness, you talked about this kind of like essentially arousal component. There was a paper from 2015 from Sepeta Saidagiani, which showed that you see essentially a more integrated network state, whole brain network state in fMRI before misses than before hits in a threshold awareness task. And I'm wondering if connecting that with the work that people like Mac have been doing on arousal as moderating that brain state. Do you think that is essentially what's happening? And given kind of your work showing that you get this representation of activity kind of across the cortex, and that the prior brain state in terms of like large scale connectivity is part of what's influencing that.
Yeah, thanks for the question. I'm a big fan of Sapedi and Mac's work. And so here it's MEG. So we don't have very concrete ideas about the sources. We are pursuing that question as we speak. Here, indeed, our model is that our working hypothesis is that the non-content specific process has, given that it's correlated with moment-to-moment pupil fluctuation, pupil size, it's probably got subcortical influences from the acetylcholine and noradrenergic systems, and they may be distributed more widely. Indeed, that's our hypothesis. And the content-specific process, we are not ruling out, higher order representations, but a very, it would make sense that visual regions contribute to this content-specific process. But we think higher order regions may also contribute. These are questions that we are currently working on.
Mm-hmm. Does that answer your question, Dan?
Oh yeah, I'm not sure. Yeah, I guess so. I wasn't sure if I was still muted or not. Yeah, no, yeah, that was essentially answering my question in terms of like whether this was like the non-content specific process was essentially this modulation via ascending neuromodulatory systems.
Okay, we have another question from the audience. Anonymous attendee, I don't know if you're out there and can oops, where did it go? Here it is. And whether you can read it off or pose it. Otherwise, I'll pose it for you. I guess I'll do that. This is a question from someone who's joined a little late because of connection issues. The question was, have you worked on perceptual decision-making with awareness? If you talked about that, could you please summarize what you've said about it?
I'm not sure if I understand the question, because our tasks all have a component of perceptual decision-making. We ask subjects to discriminate orientation or answer by category of the stimulus. Yeah. But an important addition here, which I confess was very much inspired by work from Stanley Hunt's lab, is that we incorporate both objective discrimination and subjective awareness questions on the same trial. So we can, at single trial level, look at the combination of those and ask what is the discrimination performance for seen versus unseen trials, for recognized and unrecognized trials. And I think additional innovation we did, small innovation, is that we included scrambled images, and which have been included in some of the previous studies. But it turned out to be quite important for understanding how neural activity, for example, stimulus activity influence perception. And it allows us to ask whether a specific type of activity predisposes the subject to answer recognized more easily, regardless of what's coming in, or does it potentially help you with your perceptual sensitivity in the sense of separating the increasing the distance between neural representation of signal versus noise.
Very cool. Thank you. Right. What other questions do we have from either the attendees or the panelists? Anything else out there? We've gone all the way from consciousness to control and cognition back to consciousness. We've actually made a roundabout journey around all the topics of the meeting.